Easier. Okay.
Okay, well, everybody, we're being recorded. This is PAO, which is Plan Ahead Ohio, along with OWR, Division of Forestry. And we're going to have a wonderful conversation with Jim Chatfield from OSU Extension, and he's going to be talking about plants and diseases, and then he's going to entertain questions. But before we get started, let's go around the room, introduce yourself, and tell me one of your favorite trees. Okay, well, I know we have a lot, but just give me one. Let's start with Gary.
Gary Davenport. I'm a lay minister with the Unitarian Church. I work with the Kentucky Land Bank. And my favorite tree, I was in Vinton County at Zaleski State Forest, and the thing was packed full of white oaks with brown leaves. And that's my favorite for the last few weeks.
Awesome. All right, let's move on down to Adam Lee. I see you have a guest with you. Would you go ahead and introduce yourselves?
Yes, hello. I'm Adam Lee. I am at Youngstown Cityscape, and we were talking earlier with Lola. I was introducing my friend Jaywan here. He has some interest in trees and arbory, I guess is the best way to phrase that, Lola. I don't know.
Arboriculture, arboreums, yes, that's fine.
Yeah. Anyway, so she invited us to come and can't actually stay too long. Dinner's almost ready, but we wanted to meet Jim and recommended that. Anyways, so I can pass it off to Jay here.
Fantastic.
Hi, I'm J1 Madison. I will tell you first off that I lack experience in everything, but I'm eager to learn what I can learn, and I'm excited to learn. So thank you guys for having me. Seriously.
Well,
Great.
thank you. All right. And you know what, Jim, if you wouldn't mind, he would like to get in touch with you at some point in time. So I'm going to share your contact information with him. Okay.
Of course.
All right. Let's move. Great. All right. Thank you. Let's move on to Mary June. You want to say hi?
Hi. My favorite tree is, there's 3 of them out back. Right now they have no leaves whatsoever. And I have been told by various people that they're wild cherries. They have no cherries on them, and they're huge, huge trees, and they're beautiful. And I love them.
All right. Thank you for reminding. We need to go back to Adam and to J1. What's your favorite trees?
Sorry, my favorite tree is the cherry tree because I love how it smells and the redness of the tree.
Awesome. Adam?
I think my favorite's probably probably the red bud because it reminds me it's signaling summer is coming.
Awesome. Thanks for sharing. Let's go to Diana. Come on, Diana.
Of course, I love my magnolia. I guess it's a Chinese magnolia out front. I just love that tree. Both of them.
Live.
And I love the way they give shade.
Presentation.
And I love, this is O'Doul's. It's non-alcoholic for all of you who might object.
We're very sorry. We're very sorry that it's non-alcoholic.
Me too. And anyway, then I also love my dogwood. I love pink dogwood.
Diana, you know it's supposed to be 1 tree.
Oh, I said we have.
No,
Okay.
Tell us who you are, though.
Lola,
who are
she's
you,
causing
Diana?
good trouble. She's causing good trouble.
Who are you? Tell us who you are.
I don't know. Who am I? I'm this 68-year-old lady who has a house, who's finally semi-retired. I mean, I just do work 12 hours a week. That's it. And that's doing childcare, so it's a breeze. And I just have so much fun 90% of the time, well, maybe 80% of the time. Why exaggerate? And just being retired and doing what I want, and I can just tell the world, go to.
Thank you, Diana. She's also the secretary of PAO.
68 is very young.
Secretary of the PAO. Yes. My apologies for not remembering.
68 is very young, Diana.
Right?
Thank you.
Very young,
Very
68.
Yeah.
Let's move on to Chris and Sean. Could you introduce yourselves for us?
Hi, Chris Black, Sean O'Toole. I know everybody. Hi, folks. Good evening to you.
Good evening.
Oh, yeah, I forgot who I am. POA board member and Fairgreen gardener for about 10, 12 years, community gardener. Sean, you want to say who you are? You're Sean.
Mushroom man. Yeah.
Who am I?
The
This is the
mushroom
age-old
man.
question. Well, I'm a retired property manager, and I guess I'm a master gardener. I'm a composter,
Mushroom gardener.
mushroom gardener, forager, environmentalist. I can't think of anything else that I am. I know there's a lot of
What's
people
your
that
favorite
say I'm
tree?
other things.
What's
Now,
your
my
favorite
favorite
John?
tree is the oak, which in the Druid lore coincides with my birth month. And also, some of my favorite mushrooms grow around oaks,
Awesome.
like chicken of the woods, chanterelles, things like that. Of course, I also
Chris, did you
like
tell us
the,
your favorite tree?
tulip popular because that's where most of the morels grow. So,
Thanks
My
for
favorite
adding
is,
that.
I love paper birches. That's my favorite. They're
Okay,
just beautiful,
awesome.
unique, delicate, papery trunks and gorgeous yellow leaves in the fall.
Thank you for sharing that with us. Aaron, how are you?
Hi,
I'm
Oh, Aaron,
Hi, everyone.
good to see you.
Yeah, good to be here.
All right.
Good to have a night off. Yeah.
Tell
So
us.
I'd say favorite tree, I just have to pick one, right?
Yes.
All right, so it's weird. I have a calling to pawpaws right now. I have a bunch of seeds in my refrigerator that I'm trying to germinate. So yeah, I'm gonna see how that goes, but I think it's gonna be the pawpaw. I'm gonna go with that one.
Great
All right.
tree.
If I have to do one.
Tell us a little bit about what you do.
So I'm also on PAO's board. I'm a board member on the board, and then I have the Calvin Center for the Arts. Most everybody here I know. Yeah, I make enzymes. I'm an environmentalist. I'm a big proponent of changing our lifestyle and the ways we interact in the world to make a difference environmentally and on a personal level. And then what we do
Great.
is businesses and everything else. Yeah.
Thank you so much. Hi, Don. You want to tell us who you are and your favorite tree?
That's a good question. I really don't have a favorite. I guess if I had to pick one, I'd say the mighty oak tree. But I've got a few years on you, Diana. So you're not the oldest one here, believe me.
I know that. I know that.
My claim to fame is I'm a board member with the Friends of the Mahoning River.
Awesome.
Yes.
And I'm starting a wildflower garden in my backyard. So I'm very
Okay.
interested in learning more than I've ever known.
Right. Well, you've definitely come to the right place, and I'm so glad that everyone has joined us tonight. And Jim, if you're ready, does Jim have the ability to share his screen?
Now, Lola, we did not hear.
Oh, me, yes. I knew you were going to say Lola.
Oh,
Okay,
yeah.
again, my name is Lola Lewis. I am the Regional Urban Forester with ODNR Division of Forestry. My favorite tree is the willow tree. I love the way the leaves blow in the wind. And it just reminds me of being calm and peace. So that I am the willow. Now,
All right.
here is Mr. Jim Chatfield.
I assume
And your
you're seeing
favorite
my.
tree, Jim? What's
I'm
your favorite
hoping-
tree?
I'm going to say for today, I'm going to say Katsura tree. So it is a non-native, and I'll try to show you some pictures of it if you're not familiar with it. It's an Asian tree and incredible fall color. And one of the wonderful attributes of the Katsura tree is that in the fall, and it's a little bit of a mystery as to what kind of conditions. I used to think it was wet conditions. Sometimes it's dry though. But sometimes when I go out in the morning, I have 4 little big Katsura trees. I guess they're 50 feet tall now next to our house. You get the smell of what people will say is creme brulee. Others say burnt sugar. Others say marshmallow and strawberries. A wonderful little, very, very faint but intense aroma. that is just a tiny little nuance. And I have to admit that what I love the most about trees, not only is their importance to our environment and just all their overall beauty, is just some of the little nuances, the wonderful little aspects, like the pawpaws we just harvested and made into a little pawpaw pie. So it is those little nuances, and that really neat little spicy aroma of Katsura trees is really cool. Let's see if I can, let me shift off of here just a second. Since I mentioned Katsura tree, let's see, what do I have to do here? I would have to go to pictures, Katsura tree.
How do you spell that?
Yeah, I'm going to show you here.
Okay.
So Katsura tree. And the thing I want to point out here is Katsura tree, the scientific name for Katsura tree, the Latin binomial, is Cercidophyllum japonica. And there's an importance to that. So Cercidophyllum or Cercidophyllum, phylum means, or phyllum, P-H-Y-L-L-U-M, means leaf. And Cercis is the genus for redbud. So we have a redbud lover crowd. We all probably are. And so Cercidophyllum really literally means that their leaves are a little bit like redbuds, a redbud-like leaf, and the japonicum indicates that it is from Asia. Just a really nice tree. As far as I can see, we haven't seen it become invasive, but you never know. It's been around for a long time. There's some that are over 250 years old at for example, at the Morris Arboretum at the University of Pennsylvania. So, you know, they've been around for a while. They don't seem to be invasive. Katsura trees are really quite beautiful. Let's see what happens when I bring them up here. So one of the things I liked this year was the Katsura tree color at a certain time was this yellow here. So these are the Katsura trees. The contrast here is what I liked. It was that red blueberry. So blueberry is high bush blueberry is just an absolutely gorgeous edible landscaping tree because they are really pretty in the fall. And there was the Katsura tree in the back of it. And this was like last weekend. So they've mostly fallen off by now, but still getting the aromas from the fallen leaves. This is another look to it. And this was, this is in our landscape. So some of the early leaves that we're coloring, you get this really salmon and apricot kind of colors, really quite beautiful. And here is in Scotland. And so what they have at the Botanic Gardens in Scotland, the kind of apricot kind of the fall foliage. So you can see here the heart-like leaf of Cercis, like redbud. They're not related to redbuds, but that heart-shaped leaf is just quite beautiful. This is also in Scotland. Again, just you can see how glorious it really is in variations of its fall color, from that yellow all the way to this kind of wonderful pink colors. I mean, just spectacular over in Scotland last October, I guess it was late September. Now this picture here, I want to show you because it kind of leads us into a little bit in our part of our discussion in our of today that we're going to focus on diseases. Is we're really going to focus on infectious diseases. And so infectious diseases caused by fungi, bacteria, viruses, those kinds of things. And what's interesting about this, and this is from some weeping Katsura trees at Seacrest Arboretum in OSU's Arboretum in Worcester. And this is what you would expect a viral disease, a mosaic virus disease would look like on Katsura tree, except it isn't. And so that's one of the difficulties that you have with your physicians relative to infectious diseases, and also with plant pathologists, plant doctors. Symptoms can sometimes be difficult to identify and to diagnose whether the symptoms connect to something, because that kind of symptom is really a viral-like symptom. It's little blocking off little areas, little patches, but it wasn't. This is what frost injury, believe it or not, looks like on Katsura tree. And so it sometimes goes up, please, but we'll see this from when frost injury occurs in the spring, and then it goes all the way through the season, becoming more and more difficult to diagnose once people aren't even thinking of frost injury, but it's just differential damage to that tissue. So diagnosis is an issue with with plants when we think they might be infectious diseases, but they aren't. But Katsura trees, there's the little Katsura trees on the female. They are dioecious, 2 houses, males and females on different plants. And so this is in my yard. And as you're starting to see with these colors, they come out in the spring with a rosy color, and then in the summer, they become this blue-green, and then you saw the kind of fall colors that you have. There's a weeping Katsura tree. You see this kind of thing that can happen in the spring as they're early coming out. This is 5/22 this year, so May 22nd. So they're just a lovely, lovely, lovely plant. Now here is a really neat one. So here was frost injury, and I love this picture because it shows how chimeric injury can damage. I mean, you look at this twig and you say, oh, geez, something happened there, and it was frost injury. But for some reason, this particular area of the twig, it just wasn't as affected. I mean, kind of bizarre. And you know, here you have on one side of these, they're called opposite or sub-opposite leaf arrangement. And so sometimes it's a little off center here, but this is opposite leaf arrangement. Look at that one. I mean, this one was blasted, this one wasn't. I mean, it's kind of an interesting thing we saw. And there was where you see the more classic thing you would expect to see of frost injury on Katsura trees this spring. And then in spring, that's what that little pattern was. And again, most people would say, oh, that must be a viral disease, a mosaic pattern from a viral disease. But the symptom is belying the fact that that isn't what's happening here. But again, you see the different colors there, the spring colors of Katsura tree. Just a lovely, lovely, lovely plant. I love Katsura trees. There's its flowers, you know, very cool little flowers. You don't ever notice them much, but big weeping Katsura tree. Again, I could go on and on. You can see all the different colors. You can see why people like Katsura trees. This is when you put Katsura trees. And so this kind of leads in as well. I wasn't planning on doing any of this, but this kind of leads into, we are going to focus today on infectious plant diseases, unless your questions are not about infectious plant diseases. In other words, ones that have a biotic agent, a living organism, or in the case of viruses, a non-living chunk of DNA and protein, deoxyribonucleic acid and protein, or RNA and protein. But here is an example of a non-infectious disease, so a disorder that occurs on the plants that is really not associated with an infectious organism, in this case just scorch, because the plant was planted in a bad location, damaged, you know, really, really tough thing. And so we would call this a non-infectious or an abiotic as opposed to a biotic disease. We're not going to focus on those today because we talk about those all the time. This is where we're just going to focus on infectious disease today, unless you force me to do otherwise. So Katsura trees are a delight. All right, I can go on. It's just some of my pictures of Katsura trees. Now we will go back to our deal here. All right, so I kind of put here, so Lola's plant disease lessons for 2020. Oops. Rose black spot, so those black spots, that would be a symptom, but it's caused by a fungus. So we usually will say that most common kinds of infectious diseases are caused by fungi, and that's maybe true. It's certainly more common to see fungal diseases than, let's say, bacterial diseases. But in reality, it may very well be that there are more viral diseases of plants than there are fungal diseases of plants. Oftentimes, we don't know about the viral diseases because since they're submicroscopic, we have to have had some real sleuthing with electron microscopes, for example, to figure it out. This is tobacco mosaic virus, which also gets on related plants in their family. The nightshade family, like tomatoes and potatoes, will also get tobacco mosaic virus. And tobacco mosaic virus was the first virus that was ever, I mean, at first, I don't know if we should even say the word identified, but all the way back in the 1860s, people had extracted, they had these bacterial, these filters that would filter out bacteria, which is about a micron in size. And then something came through that was also capable of causing a plant disease. And they said, well, what is that? And so it's, you know, the whole thing with virology, which we're all tied up in now with COVID-19, the whole thing with virology started with a plant virus. That was the first virus that was kind of imagined, contagium vivum was what it was called at the time. And then in the 1930s, Dr. Stanley actually won the Nobel Prize for identifying the first virus, or crystallizing the first virus, massive particles, and then identifying it. He won the Nobel Prize, but he was wrong, because what he thought it was that it was protein. So his thought was that viruses were caused by, were actually consisted of just protein. Well, he won the Nobel Prize because, you know, we still couldn't visualize viruses in the early 1830s because we didn't have electron microscopes that could give us magnification higher than 1,000 or 2,000 X. And later on, and 4 years later, after he got his Nobel Prize in 1935, we had the development of the first electron microscopes. And then, of course, eventually they realized that they were made of nucleic acid. So DNA or RNA, so the stuff that is our genetics. Of course, they didn't really know exactly what all that was about or what the structure of DNA and RNA were in 1935. But it turns out that viruses are these nucleic acids with protein caps. So they did have protein there, but they actually were not what really makes viruses work in terms of infecting people. It's that genetic material that does that, and then some other little structures on there. And then, of course, we figured out the structure of DNA in the 1950s with Watson and Crick and Franklin and Wilkins, who determined what, you know, the structure of DNA and all that sort of stuff. So, but, you know, it all started with viruses in terms of people understanding what was going on with a plant virus. And so I don't know, I might get into plant viruses a little today. As the pandemic really got us to doing a lot of online programming, I put together a program on virus diseases because we rarely talk about them generally when I do programs, unless I'm working with a grower group where a viral disease is really a big problem, like on hostas or something like that. But so I put together a program, and I learned a lot in putting together the program. I mean, I knew some stuff, but I learned a lot more. So we might talk about that a little today because it's rather interesting. And then here we have a fungus. Now, many bracket fungi are simply saprophytes, and so they're not parasitic. And for an infectious plant disease, you have to have something that's a parasite, actively causing infection inside plant cells. Saprophytes are just living on already dead organic matter, so they're not really plant pathogens. They don't have pathogenicity, and then there's things all along that scale. But this is a bracket fungus, which is really quite important. It's called let's go to Don. Don, what is this one?
I have no idea.
Well, it's a really neat one, and it has a really evocative name. So it's a polypore, and it is the common name for this, not the scientific name, but the common name is really neat. It's called dryad saddle. And the idea is that in the Greek myths, the dryads are little mythical creatures that are up here kind of protecting the forest. So they're sitting up here in their saddle, looking out for the forest. And so called dryad saddle, also called pheasant's back polypore, because it oftentimes has little spots that kind of look like a pheasant and all that kind of stuff. But it is not just a saprophyte. It is a parasite. It's a pathogen. And so if you see this on your trees and those trees are next to the swing set, then you have to be concerned, because when they fruit, because when we look at in fungi, when you see the fruiting body of a fungus out on the stem of a tree, in many cases, it means that maybe the infection with this fungus was 10 to 20 years earlier. And then the fungus was ramifying throughout that stem, and then eventually it produces a fruiting body. And so when you see this, this is telling you, there's some real problems with decay in there, and it's active because this is a good pathogen. And so you kind of see that's kind of an important thing, is something, I mean, you can have a fungus most fungi are certainly not plant pathogens. Most fungi do not cause disease, just like most bacteria don't cause disease. Most viruses, don't cause. So, but the fact of the matter is some of them do. And so identifying those turns out to be rather important. All right, here is a little Shakespeare, and I love this quote. And I altered the quote a little bit. It's all men make faults, but I put all plants make faults. So roses have thorns and silver fountains mud, clouds and eclipses stain both moon and sun, and loathsome canker lives in sweetest bud, all plants make faults. The reason I like to use this quote is to get us off the idea that disease is nature gone wrong. Disease is not nature gone wrong. Disease is when something happens maybe that we don't like. We don't like infections of plants that we want, but that doesn't mean that it's unnatural. Diseases are natural. I mean, sometimes, we can stress plants and put them into environments that aren't really ones that suit them well, and that makes them more susceptible to certain diseases. But the reality of it is that disease is part of nature. I mean, when it comes to nature, the pathogen that causes a fungal disease is co-equal to the plant that it causes the disease on. They're just organisms that are interacting. And when and it's important to keep that in mind. So we're not talking about something that's unnatural. We're talking about something that's natural. Now, admittedly, we like to do unnatural things. In fact, we all do unnatural things if we're gardeners. I mean, you might say, I just want to do whatever nature lets happen. That's probably not technically true. I mean, it's always a combination of nature and nurture in my mind. I mean, let's face it, we are not typically willing to just I threw some seeds out there and I call it my vegetable garden, and whatever came up is fine. I don't care about weeds. Whatever happens, I don't care. All I want is a natural vegetable garden. Well, usually there's not very many people that will accept all of that. They really do want to modify nature to some extent. They want to harvest a certain amount of a plant. So we always are compromising, but there is a great saying, naturam expelleth fuca tamen usque recurrat. You can drive out nature with a pitchfork, but she always returns. So we also have to be aware that, there's processes in nature that are not necessarily the processes that one of the components of nature, might want to happen in a particular case. But the fact is, all plants get diseases. There is no type of plant in the world that doesn't get any infectious diseases, any more than there's any human on the earth that is immune or does not get any diseases. That's not biologically conceivable. So the fact is, diseases are natural, but we sometimes, of course, want to manage those diseases in ways that don't cause us problem. We don't want all our potatoes to die in Irish potato famine and that sort of thing. And we'll talk about that a little bit. So I like this quote. All right, a fundamental thing, and if I don't do any more slides and we just launch into questions, I really do want to make sure we get a feel for this. There are three components of what we call the disease triangle. Now we call this the plant disease triangle, but you might as well call it the human disease triangle, whatever you want to talk about, animal disease triangle. You have to have a susceptible host, a particular plant host that is susceptible genetically to a particular pathogen. So you have to have a virulent pathogen, something that is capable of causing an infection. You have to have a susceptible host, one that is susceptible to that particular pathogen. But you also have to have an environment conducive to the disease. You don't have the right hours of moisture at the particular temperature on a rose leaf, and it will not get rose black spot. So you have to have an environment conducive to disease. And that's why rose black spot is not a problem in greenhouse rose production, modern greenhouse rose production, because you're going to have it inside. There's no rain inside, and you're not going to use irrigation. You're going to use trickle irrigation. So you're never getting water on the foliage, essentially. So you would never have black spot, because the key to that particular disease, and all diseases are different, is the number of hours of leaf wetness. So you have to have all three of these things. The presence of the pathogen doesn't mean that the disease occurs. The presence of a susceptible host doesn't mean a disease will occur. A particularly inducive and conducive environment for a disease doesn't mean disease occurs. You have to have them all three. And so that's a kind of a truism or whatever, but the power of that is, okay, if we are going to try to involve nurture to prevent a disease on a particular crop or plant that we want. And for example, and Don probably knows this, there fungal diseases of mushrooms. You know, there's a species of the verticillium wilt fungus that causes us all kinds of problems on maples and tomatoes and everything. There's a species of verticillium though, which also causes a disease on our nice button mushrooms. So fungi have disease, mycoparasites, all organisms have diseases. So at any rate, you have to have all three of these for the disease to happen. And so it's figuring out, you know, where do we have something that can allow us to not have a disease? Let's plant a rose that doesn't get black spot. Let's eliminate the pathogen with good sanitation or a fungicide. Let's modify the environment so that we plant certain plants out in the sun rather than in the shade, because so there'll be less relative humidity and less moisture on the leaves. So that's what's really powerful when you start thinking, okay, what do I have that I can use to prevent disease from happening in each of these little areas? That's a good way to then think about how you might manage disease. All right, so I'm going to stop here for a minute and say, are you ready now to just start asking questions, or do you want me to do a little bit more background? Anybody have questions that are coming up already?
Forward.
You have to unmute.
I'm not hearing any questions. By the way, I'm very pleased by the group. We have a king in the audience. That's Don Rex. He's a king. We have a goddess in Diana. I mean, think about it. We have a goddess. We have a king. You know, if we had had a more perfect union, then now I've forgotten his name. Jaquan Madison.
Yes, Jaquan.
Madison would have been putting together the Constitution of the United States, we would have been in a much better shape than when we made some mistakes early on in our you know, I mean, James Madison was a good guy, but Jaquan would have been maybe a little bit better in terms of our more perfect union that we got off to a bad start for political reasons.
Right.
So, you know, we have a lot of, you know, kind of really powerful people out there.
Yes. Chris, did you
any
have a question?
questions?
Yeah, I sent a question in through the chat on the bottom about powdery mildew.
And I can't see those questions, so
sorry.
moderate. Yeah.
Oh, okay. I thought that was our way of communicating.
It is, except the person doing the presentation can't see it.
Okay. But I had also put in the box, if you have your questions, to go ahead and ask him directly tonight.
Oh, okay. Okay.
Thank you.
So I just wondered how you dealt with powdery mildew. Every year it's a problem in my garden. I've used a copper derivative. I've used some organic sesame seed derivatives. As far as sprays, once I catch it, you know, I know that airy, keeping your plants at a good distance so that they're not crowded is a good way of keeping. But anyways, Jim, I always lose at least 3 or 4 plants due to powdery mildew. So I was just wondering how you might deal with that. Or
okay, that's a good question. Now I would like to ask a question here. So let's just make sure that we're all looking at what powdery mildews are. Let's make sure that with that question. So I'll just put powdery mildew in here and see in my pictures what I come up with. But so there's a lot of, that's a good question. And this is a good, I mean, I kind of like it when people just, you know, bring in the questions, because it allows us to really, so, you know, when we talk about powdery mildew, let's take a look here at some things.
it affects so much in my garden. You know, it goes
so
from
there's powdery mildew on elm,
yeah.
powdery mildew on a buckeye. There's powdery mildew on a calorie pear. There's powdery mildew. These are all just recent pictures I took this year. On crepe myrtle that we're now starting to be able to grow in Ohio, there's powdery mildew and
wow.
blossom. There is a picture, this is my picture. This is, believe it or not, that's what powdery mildew, if you could actually see it. And I always like to make this point that, you know, powdery mildew, the conidia, the asexual spore structures, are really interesting. They occur in chains, and, you know, and it's a good way to think about stuff. And then there's a little, what's called the hostorium of the fungus that gets down into the superficial level of the leaf, of the first layer of leaves, the epidermal cells of the leaf. And what's kind of neat about, and conidium is just a name for an asexual spore. And so what I like about using this, I don't usually use this particular slide that I happen to have in my thing there, but what is neat about it is that, you know, a person might say, well, I never see these chains. Well, that's because they really are microscopic. So that little film that you see, believe it or not, there's chains of these little spores. It's just that the thing to remember is that they're microscopic in size. We don't actually, you know, that's the difficulty of plant pathology, is the fact that our pathogens are not something you see with the naked eye. So there's
Hey, Jim, if you still want to show the picture is gone. Did you still want to show that?
You're not seeing anything?
No, we were, and it just went.
I can see the conidium and the picture of the conidium,
Yeah.
and it's infiltrating the H.
Yeah, that's what I want to be showing. What are you not seeing, Lola?
Well, it may just be my computer, so go ahead. It might be
Yeah,
just-
sorry. Are others seeing this little drawing?
Chris can see it.
Yes.
Yes, thank you. Yes, I see
Okay.
it.
So this gives you that sense of how small it is. And then there's little strands of the fungus, and then there's these little structures that then penetrate this epidermal cells. So this is another interesting point about powdery mildew. Powdery mildew diseases are usually considered to be not as serious as other diseases for this reason, is that they typically are much more superficial than most pathogens. And so in most cases, we don't think of powdery mildew as being a killing disease. Now you're describing something different, so we'll get to that in a minute. But because they don't go deeper into the cell. So look, for example, if you talked about rose, the powdery mildew fungus on rose is not as serious as the black spot fungus. The black spot fungus goes down here into the mesophyll cells and causes damage, and then you get leaf drop and all that sort of stuff. Powdery mildew is much more superficial, which is very important, because if you were to ever use a fungicide, what you have to do typically with a fungicide is that you have to apply it before the fungus arrives. Before the fungus arrives. Now that's a problem, because I just told you that you can't see it. So that's the difficulty of plant diseases. It's very difficult because you'll never see the pathogen causing an infection. You'll never see it. It's not like an insect. Insects are gargantuan compared to plant pathogens. Insects are not plant pathogens. Insects do not cause plant disease. They may vector the pathogen from one plant to the other. So that's a problem. That's a fundamental problem of the difficulty of managing plant diseases, is that you can't see the infections. The pathogen is microscopic. Now you see some structures that are macroscopic, like that bracket fungus that we saw with the dryad saddle, but you never see the infections. And so, you know, all right,
Jim,
decide to use the fungicide before you see anything happening. Now, I'll take a question here in a minute, but so typically their powdery mildews are easier to control with fungicides because they don't penetrate deeper. And we're not good at all of getting anything that gets down in here. We're just not any good at it. We're not able to control diseases once they're down in here. Now you can kind of control a powdery mildew fungus that's just in that epidermal layer of cells, which is all the further that it gets. But the other part of this piece of this puzzle is that for the most part, powdery mildew fungi don't kill anything. They block out photosynthesis to some extent. Usually what we talk about with powdery mildew fungi is that they reduce, I guess I need to take a little sip, is that they reduce yield. So for example, years ago, people felt that you could not possibly get enough money out of an acre of wheat, you know, because it's per bushels, it just isn't that high value of a crop compared to, say, roses. I mean, it may be more powerful for humanity, but you just can't. It used to be felt like the cost of using a fungicide for control of something like powdery mildew of wheat was more than it would be worth by making applications. Well, but then some people came up with some powerful fungicides. They came up with some techniques and some timing, and they would end up with yields that were 20 and 30 bushels more per acre if they controlled powdery mildew. And this was in Britain. And so, okay, so that's kind of the cost benefit consideration that people have for these kind of things. And you're able to make applications after the fungus has already infected the plant with powdery mildew, where you can't really do that with other plant pathogens. You have to just make the assumption with timing relative to what kind of environmental conditions occur, but that's the difficulty. All right, so then let's go back to your question. Well, one thing before we lay out and delve into your question. This is an interesting aspect of it. The powdery mildew fungus that gets on lilac is not the same one that gets on wheat. It's not the same one that gets on rose. It's not the same one that gets on turfgrass. It's not the same one that gets on zinnias. It's not the same one that gets on apple leaves. It's not the same, and on and on and on. And that introduces a very important issue associated with plant diseases. The host range is critical. Most plant pathogens have narrow host ranges. Not all of them. The verticillium wilt fungus has a very wide host range. The gray mold fungus has a very wide host range. But most plant pathogens have a very narrow host range. And powdery mildew fungi typically have a very narrow host range, one species or one genus of plants. And that's important because then that tells you something. You could go to a rosarian's program, somebody who is very proud of their roses, and they know that they're using roses that are susceptible to powdery mildew because they want to have those particular roses horticulturally. Instead of using mildew resistant ones, they just have to have certain cultivars, but they spray religiously and they're very concerned and they want to do something for their roses. And you could bring a bunch of lilac leaves that have powdery mildew all over them and rub them on their roses while you're laughing at them at your party. And they may hurt you, but they would be wrong to do that because you're not really transmitting powdery mildew from this lilac leaf to their rose. The lilac powdery mildew fungus will not infect rose. They are highly specialized fungi, even though they look the same. All right, so having given that little bit of a background, what plants do you have powdery mildew on and which ones of them died from powdery mildew?
Well, first I find them on my squash, zucchini.
Yeah, it's
in
pretty bad on
particular.
squash. Yeah.
Yeah. And I usually lose a few of my squash, mostly zucchini. And then I find it on cucumber, anything that, the trellis plants, the climbing plants. And then I do have it on bee balm. And lastly, zucchini. So I was assuming it was the same kind of a fungus, but according to what you're saying, no. But the other part of my question that I wanted to ask is, does it stay in the ground? I mean, you already said that there's really nothing that you can do once it goes below the surface, but does it, is that something, would crop rotation help?
Okay, so you've introduced some very important things. First of all, the zucchini, the squash, the cucumbers, those are all cucurbits. So those are all very closely related plants in the Cucurbitaceae family. So they're very closely related. Now there are several different powdery mildew fungi that occur on those plants, but there very well may be a little cross infection because they're very closely related plants. The one on bee balm is completely different fungus. Those are all plants that can get a lot of powdery mildew and make them look pretty ugly. I mean, I still would say that I rarely see them killing those plants, but you can get to the point where you certainly ornamentally killed the bee balm. I mean, it's so ugly that you don't like it anymore. And it's a late season kind of thing as the squash and the zucchini is starting to decline. So, it can be pretty bad on those plants, no question about it. Now, the other thing you have to bring into the equation before I come to some things that you brought up that are also very fundamental, that's great we're getting out right now. But the other thing you have to be aware of is that there's another group of pathogens called downy mildew organisms. We used to call them downy mildew fungi. We kicked them out of the fungal kingdom and now we think of them as water molds. But downy mildew is a much more severe problem. Downy mildew will kill plants. And when we get downy mildew on cucurbits, it's a big alert that goes out every year for our growers, because if you get downy mildew, which is also has kind of, it's downy instead of powdery. It's a little bit darker. It's a little bit more grayish. It tends to be more on the undersides of leaves. You tend to get a little bit more angular scorchiness on the upper surfaces of the leaves. So we would have to figure that part out. if you're getting downy mildew, that's a significant issue that's much more severe. But one of the things you brought up, which was very helpful in our discussion, is one of the keys to understanding how to manage disease is the disease cycle, the cycle. And one of the, when you first have infections early in the season, does the fungus produce a second set of spores? Does it have different kinds of asexual and sexual spores that do different things? And a very important question that you just asked, how does the organism that is the pathogen overwinter? How does it make it through the winter? Probably with most of our powdery mildew fungi, it's in crop debris, and it also is in buds. And so with with roses, for example, it's going to be on the buds. With apple trees, it's going to be on the buds. With your cucurbits, it's probably going to be in debris that's in the soil or debris that's in your neighbor's soil of the plant. So it's one of the reasons why sanitation at the end of the season can be helpful. Sanitation is a very important component for disease management. I would say that not so much with powdery mildew fungi, because there's going to be so much of that around that it's going to be hard to do a good job. And you're not talking about perennial plants, but sanitation is critical. For example, if you have rose black spot on your plant, you want to gather up all the black spotted material at the end of the season and get it out of there before the next season. If you have peonies with peony blotch, which are a couple of different diseases that occur on peonies, you want to cut those plants back and then remove all that foliage and get it out of that planting, because it's going to be the overwintering inoculum that's going to get things off to a roaring start the next season. With powdery mildew, other than just cleaning up the debris, which can help you over time, you're going to be dealing with powdery mildew on your cucurbits every year, in my opinion, unless you decide to spray. And you can spray effectively. It's just a matter of what you're going to use. And you're using what?
Recommendations for use. I've had copper derivatives. I used inorganic spray this past year with sesame seed oil. And I think there was fish oil in there as well. I mean, they all do work in time, but it seems like a lot of the leaves are destroyed, and some plants are just hanging on to dear life once you recognize that powdery mildew or downy mildew is there. And it just seems to really overtake the plants very quickly.
So once again, and you're just doing a great job, Chris, once again, you brought some really salient points. First of all, you're going to have to be scouting. One of the most important components of disease management is scouting. You can't wait until the disease is present and then decide to do something about it. I mean, the fact is you have a little bit of a window better with powdery mildew fungi than you have with other fungi, but still, you just can't wait until it's a big problem and then decide to do something. That may be what you can do with insects, but with plant pathogens, you have to stop the organism from getting into the plant. A little bit of room for not being right at the moment with powdery mildews, but you got to get it early. So that's really pretty important. Secondly, I would say that copper sulfurs can be helpful. Coppers are going to be a problem if it gets cool, then it's going to damage leaf tissue. Sulfur is going to be good if it doesn't get too hot, and that's going to damage plant tissue in addition to working for the fungus. And the sesame oil and the fish oil won't do anything. So you have to make it, I mean, it's just the way it is. And then there are kind of fungicides that are available.
I have to ask you one more quick question. Do you remember Jerry Baker? He
Sure.
was the guy that had, all kinds of, he used to use Coca-Cola. He would use vinegar and all sorts of, household products to combat stuff in the garden.
Right.
One of his recommendations was Murphy oil soap.
Love this.
Uh-huh. So
I don't know, just wondering what your take on that is.
well, so Murphy oil soap doesn't work for powdery mildew. So
Okay.
here's the story with Jerry Baker. Sometimes he would suggest certain things that could be helpful. I mean, why would he be wrong all the time? But there's a really good story for Jerry Baker, and it really is salient in these common days of fake news and situations we're in right now. Fred Hauer, who was a really good horticulturalist, was once on a program in Detroit, and it was like one of the garden shows where they would bring in the media, and you'd have a little, you'd be interviewed and have a little, so it was a little media show of the, you know, the Detroit Flower Show or whatever it was. And so Fred gave his talk, and then as Fred was standing by with one of the producers of the show, and Jerry Baker came on to give his thing. Jerry Baker was from Michigan, very popular, and so Jerry Baker launched into his, you know, mix up birth control pills, hydrogen peroxide, and whatever, and spray it on your lawn, and it'll control all the insects in your lawn. And so he would have all these home remedies. Now, there's a couple of things that happened there. First of all, people love that stuff. People love that idea. They don't, it's really part of the whole thing of, oh, these stupid pointy-headed intellectuals, they got all this stuff, and just use what's in your, way. My grandma used to use birth control pill or whatever. And so one of the things, so the producer said, well, you're looking a little skeptical of what Jerry Baker is saying, and so, Fred mentioned, there's not much evidence for a lot of what he's saying, but it's highly popular. And so, so the guy comes off stage, and Fred had no idea that the producer was going to confront. He says, well, this guy here says that there's no reason for most of these recommendations you're making. There's no scientific background. And so Jerry Baker said to Fred, I have about last year I had about 995,000 reasons for what I say. That's how much he made. With like America's Master Gardener and his books that Jerry Baker said. Jerry Baker knew that people wanted those kinds of home remedies. He knew that when people ask questions about a problem, what they want is for you to tell them a specific answer. Well, what you do is, 3 drops of hydrogen peroxide, a ground up birth control pill, and and 7, flakes of ivory soap. People like that. And he knew that. And he sold that idea that people like simple, they don't want all this baloney that scientists will say, or the person would say, well, I'm not sure what you're seeing there. It could be this or it could be that. That's a dilemma. Somebody, my houseplant died. And, your answer may very well be, it could be a watering problem. Well, what kind of a watering problem? Well, it could be too much or too little. Nobody wants to hear that. What do you mean too much or too little? Tell me something else. Tell me that it needs 6 drops of this and 10 drops. And so the fact of the matter is, you don't have to, listen to me with that analysis of Jerry Baker, but, let's just be honest. People like you to give a direct, absolute, quick, pithy answer. They like that, and he harnessed that.
Mm-hmm. I lost you guys somewhere.
All right.
What do you mean, Chris?
Oh, wait a minute.
There's powder.
I lost video all of a sudden.
Oh, I don't know.
We can see you. We can see you.
Okay, good. I mentioned Jerry Baker. That's like, ooh.
Yeah. All right. So, here's powdery mildew on oaks.
Right.
Can you see it, Chris?
Not yet. No, I don't know what happened that we lost video all of a sudden.
It'll come back.
Yeah.
So can you not see my pictures?
Here we go. There we go. Yes.
Yes. Now they can.
Thanks for
There's a nice little drawing here with a stomate air, the gas area, and then the little powdery mildew fungi. There's on rose. There's powdery mildew on a sycamore, a London plane tree, rather. So you can see that there's powdery mildews on all these different plants.
Mm-hmm.
It almost gives it a really white appearance. This is down in Kentucky on some crepe myrtles. And then here, you see these little black structures here?
Mm-hmm. What about them?
Those are the fruiting bodies of the powdery mildew fungus. So this is late in the season. And so what we're looking at here on your favorite tree, Lola, this is a willow leaf, and it gets powdery mildew. Here's late in the season. And so you see the powdery growth, which is the strands of the fungus plus those chains of spores. And then in the fall, most powdery mildew fungi will mate. 2 strains will come together, 2 sets of DNA or chromosomes, and they then produce a little fruiting body. In this case, it's a cleistothecium, which means it's a little closed ball. And you can see those cleistothecia barely. You can't see individual cells of the spores. You can't see individual cells of the strands. You can't see individual cells of the cleistothecium. But you know, it's big enough when you have a fruiting body of the fungus that you can see those little structures. So, you know, just, you know, you can see, I mean, it gets on so many different, these are just pictures that I've taken over the years, all the little powdery mildews that occur on different plants. All right, so I'll get off that if you want. Any other questions?
What causes it?
So what causes what?
Powdery mildew.
It's an organism. What causes humans?
Okay.
What causes an aardvark?
Oh,
I think what she's asking is what conducive environments and what is the conducive environment for it and what kind of host? Okay, you've told us the host. What is the environment that makes, I mean, it's not going to grow in the Arctic, is it? Not during the winter. What?
Okay, so very good. So the fact of the matter is very good clarification and a good point.
yeah.
As it turns out, I'm not going to do a Jerry Baker. I'm going to tell you that it's a mixed bag. There are powdery mildew fungi. Powdery mildew fungi in general do very well in high relative humidity environments, but not all of them. So, for example, there are powdery mildew fungi that do very well in the desert. So there are, and so this goes to the fact that that virulent pathogen, there are all these different fungi that infect different plants. They're different. By the way, they're not the same as the mildew on your shower curtain. So, and they're not the same as downy mildew organisms. So the fact is that there are a whole wide range of them. I think we can generally say that typically powdery mildew conditions conducive to environment are high humidity, but not always. Now that does bring up.
I've always thought, but
What's, yeah, now let me read.
That's when it always hits my plants is during the high humidity.
Right, so that's the usual story, but let's go some down here. I like the way you guys are doing this. Let's see if I can bring this question up. This is a really good question about now we're going to start with something else. We're going to start with black spot, but you're going to see in a minute. So there's rose black spot.
Mm-hmm.
You see the yellowing that occurs, the black spots. It can be a pretty severe disease year after year. I mean, it can mean that the plant just becomes debilitated over time.
Okay, year after year.
Because the black spot fungus survives on the canes, it survives on the leaves that are not taken out of the planting. So sanitation is very important.
And whether it does it develop spores so that it can last through winter or what?
Yeah, the fungal fruiting bodies survive on the plant tissue over the winter. And the strands of the fungus survive on the plant bodies over the winter. But let's address this issue that was just raised by, I don't know who raised it. The question about what are the environmental conditions conducive to powdery mildew, but also rose black spot. And so I always like this example. I got this out of an annual review of plant pathology many, many years ago. And I think it's a really good example. So prior to World War II, so trying to follow, this is kind of an interesting story. Prior to World War II, rose black spot was worse than powdery mildew on greenhouse grown roses. After World War II, powdery mildew was worse than black spot on greenhouse grown roses. So true or false, the answer is true. Okay, so then the question becomes why? What would World War II have to do with it? And it's a really good story. Does anybody want to give a try as to why that would be?
Nuclear reaction?
It's true. Now, I'm sorry, what?
I just said nuclear reaction, but it did.
No, nuclear reaction. Yeah, so it wasn't Hiroshima and Nagasaki.
No, it wasn't. Okay.
The insecticides, wasn't it? After the war, they ended up coming back with all these different insecticides that became popular post World War II. Correct? Like, isn't that DuPont took stuff from Germany?
Okay, so you're very close. So that you're really on the right track here. But what would insecticides have to do with it?
They had to delouse the troops. Who knows?
But I mean, what would insecticides have to do with these fungal diseases?
Insects spread them.
Oh, the
No, I mean, that's a really good idea, because insects are very important in spreading many pathogens. They don't spread disease.
But
No.
they work like, aren't they almost symbiotic environments, right? So they're creating that environment that would necessarily work well to create them, like the sooty mold and like different insects work well together. So if you get rid of the insect, then you sort of have more control over the
These are all really thoughtful. These are all really thoughtful and interesting ways of looking at it.
DDT.
Or plants imported from Europe.
No.
Came back with the dirty uniforms.
Dirty uniforms.
There was,
Well, you know,
chemical warfare agents.
Warfare agents. All right, so you're nibbling around the edges. Really wonderful. I mean, it's really good. So
insects weaken the plants.
yeah, and that is sometimes really a key, is that, you know, a pathogen might be opportunistic, coming in on an already compromised plant. There's a lot of fungi and a lot of plant pathogens that do exactly that. But that's not the story here. But
Did they mutate?
They didn't mutate, though that, of course, can be the case. I mean, you might have had new rose cultivars, and you could have mutation of the plants. You had different cultivars of the plant. You could have had different evolution of different strains of the fungi that are causing the diseases. All those things are possible.
Wait, can we
Here's
just
a-
ask a question?
Insecticide cause more like moisture problems.
You're right there. Now, it was a miticide, as it turns out. So if you grow roses indoors, so we're talking greenhouse here,
Yeah.
spider mites are a big problem. They used to be a hugely difficult for production of greenhouse grown roses worldwide. Now, in World War II, one of the things that happened, for those very reasons that you guys have already brought up, disease is a huge problem. I mean, if you ever read a really neat book called Rats, Lice, and History, Rats, Lice, and History by Hans Zinsner, written in like 1939, maybe a little bit earlier than that. And at the time, what he said was, I don't know if it's still quite true, there was so much mass slaughter in World War II in so many different ways. It may have changed, but before World War II, what he claimed, and most people accept, is that more people died in warfare from arrows and bullets and everything else. More people died from infectious disease, human diseases.
Yeah.
And so that, rats and history. So, these rats and lice and all these things are vectors for the pathogens that are killing people, the disease-causing organisms that kill people. So, it is true that, I mean, they did dust soldiers with DDT, and they did all kinds of things because they recognized that these soldiers, under such incredible duress, were going to get infected by these pathogens that were being vectored by these insects. So that became a huge issue. Well, as you also know, a lot of stuff came out of World War II from chemical companies, and certainly from some German chemical companies as well. In this particular case, they developed powerful miticides. I mean, that wasn't their intention, but the products that they developed for use for biological warfare and for protection from biological warfare, the products that they developed, some of them turned out to be very powerful miticides. So it turns out that so the one other little piece of the puzzle that we need to introduce here, I've alluded to them a little bit, but not in any way that would make sense. Rose black spot, for the pathogen, for the fungus that causes rose black spot, the issue in terms of whether an infection is going to be successful, if the pathogen is there and the susceptible host is there, is the number of hours of leaf wetness. And the number of hours of leaf wetness that are required for the fungus to germinate and the fungal spore to germinate and penetrate the plant tissue varies with temperature. So, at different temperatures, it's a different number of hours of leaf wetness. But the key is leaf wetness, not high relative humidity. It's leaf wetness is the key. And we actually have charts. I mean, we have charts of how many hours of leaf wetness are required for the rose black spot fungus to infect rose leaves. Powdery mildew of rose, and remember again, powdery mildew of rose is different than powdery mildew of lilac, et cetera, et cetera. powdery mildew of rose happens to be a fungus that cannot tolerate hardly any time in free water. It isn't a organism, it isn't, those spores don't do well in free water. It's not like water is poisonous to them. It's just that they don't survive long in free water standing on the leaf. They thrive on high relative humidity. That's one of the powdery mildew fungi that does well under high relative humidity as opposed to the ones out in the desert. So the powdery mildew of rose fungus, Spherotheca pinosa, is a powdery mildew fungus that does well in high relative humidity, but it dies fairly rapidly in free water. So here's what happened. Those miticides that came out of World War II, that wasn't what they were trying to create. They were looking at insecticides and other kind of uses for the chemicals, but they developed very powerful miticides. And so where spider mites were a terrible problem in greenhouse rose production prior to World War II, after World War II, mites just weren't a big problem in greenhouse production of roses. They weren't a big problem because they sprayed regularly and they controlled the mites with chemical miticide sprays. Now, how did they control mites before World War II in greenhouse roses? What do you think? They didn't have effective miticides. How do you think they controlled them?
Water.
Water. They sprayed multiple times of the day. Many times of the day when they had a big mite problem, they sprayed coarse water on the rose plants in the rose greenhouses, washing the mites off. And in fact, you can do a pretty good job of controlling mites on plants if you continuously wash the mites off. The rose wand, which is a type of wand that you put on the end of a hose to spray, was a coarse spray. They would just drench those plants multiple times a day. Well, guess what? That was a avenue for rose black spot to be a big problem in rose greenhouses. They're washing those mites off, and eventually the mites get tired and don't crawl back on. So they controlled the mites with the water, but they made it so that there was water on the leaves constantly in those greenhouses that had mite problems. And so rose black spot, that fungus loves those conditions. But the powdery mildew of rose fungus does not love those conditions. The powdery mildew of rose fungus loves high relative humidity, but not free water on the foliage. So it all switched because of the environment. So this to me is a great example of how the environment conducive to disease comes really into play here. You changed one practice, the control of mites in rose greenhouses. You changed the environment, you changed which disease is a problem. And so currently we never see rose black spot in rose production because, again, they're not spraying water on the foliage. They're not watering that way. They're using trickle irrigation. They don't have rain because they're growing in a greenhouse. But powdery mildew, because it's a very humid environment, is a problem across the roses that are susceptible to powdery mildew, which are pretty much all of them. So environment conducive to disease, which was that original question in terms of why is there mildew on that plant? What is it? Why is it there? It's there because of the environmental conditions are absolutely crucial. So what is the take-home message there relative to your management of roses? Well, there's not a lot of the basic take-home message is this is how important environment is as to whether there's going to be a disease. You absolutely have to have the environment conducive to disease or the disease won't occur, even if the other two components are there, even if the pathogen's there, even if the host is susceptible, you have to have an environment conducive to disease. Now, if you're growing roses outside, it's not that encouraging to know that particular piece of the puzzle relative to the idea that, you can't build a greenhouse over every one of your roses. And so when are you going to have problems with these diseases? You're going to have problems with these diseases in wet seasons for the rose black spot and in high relative humidity seasons for the rose powdery mildew. But so it doesn't necessarily help you manage that disease absolutely, but it does help us understand how disease works and how much importance there is to environment conducive to disease. Now, of course, part of this issue of this whole example is that from a practical point of view, it's irrelevant, for example, in the United States. We used to be major producers of greenhouse roses. I mean, in Barberton, Yoder Brothers had tremendous rose ranges. And in New Philadelphia, we had big rose ranges in Ohio, and we certainly had huge rose production and cut flower production in the United States. We simply have been wiped out by three components. We've been wiped out by cheaper labor and cheaper energy cost in South America and Africa. And we've been wiped out for greenhouse flower production, by the efficiency and marketing and quality control of the Dutch. There's no American exceptionalism when it comes to cut flower production in the United States compared to these other countries. All right, I see you pondering.
So do you have more of these trivia questions that we could play like games with or something?
Well, we can do that with any of them, or you can create your own. Let's see. I mean, I'm trying to think of when we have a particular question. I'll try to think of scenarios in which that's the case. Let's see where we might do something like that. I have a nice little story with late blighted potato, but we'll come back to that. I like your. Well, before I kind of think about what's going on here, whether I can come up with a trivia question, let's go to this rose black spot management. So we talked about that a little, and again, how I would see that using the disease triangle is useful. So this is a little OSU bulletin. So when you think about how we manage this disease, well, okay, avoid overhead irrigation. So obviously that's a problem outside when it rains. But if you are going to grow a lot of roses and you love to grow roses, you might use trickle irrigation. You might want to use that as opposed to sprinklers. And if you're going to water and use sprinklers, you just can't do it any other way. Don't do it late in the day, because as you have a particular amount of moisture, as you go into the night, and the temperatures go lower, the relative humidity goes higher, and the potential for water remaining on that leaf, free water remaining on the leaf, increases as the temperatures go down. So if you're going to have to water, getting water on the foliage, do it in the morning as opposed to late at night. So that's trying to manage the environment. Prune away shade-producing vegetation to improve rapid drying of foliage. So you want to prune away that shade-producing vegetation that's near your rose planting. You also want to plant roses in the sun so that they don't tend to stay wet longer. So those are ways that you consider that environment component of the disease triangle and what you're trying to do. How would you manage the pathogen? You destroy infected leaves and canes from the previous year, either in the fall or in the spring before the leaves come out. That sanitation is trying to manage the pathogen. Or you could use an effective spray program with a properly labeled fungicide beginning when leaves emerge in the spring. That's managing the pathogen. Or some varieties have been reported to be resistant or tolerant to black spot. That is managing the disease by planting roses that have genetic resistance to rose black spot. So that's an example of kind of how you use those components of the disease triangle to manage a particular disease.
Okay. Now, how many people grow roses?
I don't know.
Who among us grows roses?
I do.
Who said I do?
Me.
Chris.
Yay, Chris. Okay.
I have small tea roses and they do have black spot and we're managing okay. You know, I haven't really much, you know, we just go with the flow. But I think that's really helpful when you say, Jim, to use the trickle type of watering and use it at the beginning of the day versus the end of the day. Because that's, you know, when I'm recently retired, but when I was working, that's when I had the time, was to come home from work and water my plants.
Sure. All right, Diana. So what are kind of the plants that you like to grow?
I don't. What do I grow? I mean, I don't know. I Anything that'll grow. If it grows, I keep it. You know, if it lives.
You've cast aspersions on Chris for growing roses. So
No, I have not. I used to grow roses when I had on Woodhurst. I grew roses because I had sun and very few trees, so I could grow roses.
right.
I couldn't grow. Forget it. I can't do roses here.
Yeah, your backyard is very shady.
You
Not
grow
conducive to
hydrangeas or
roses.
try.
I've got a question with. Thank you, Hope. I've got a question with my hydrangeas, but it's not a question of disease. I have greenery. I have these beautiful hydrangea plant that's huge, and it has flowers. And it has no flowers. It has 3-4 flowers the entire season. What do I do? One year it had no flowers at all. And I figured it's because I cut the canes back. The next year I left the canes and it had about maybe 10 blooms. Okay. And this is like big, you know. And then this year I didn't cut any of the canes and it had 4 maybe. It was just, I don't know. So I cannot get them to flower, and I don't think it's a disease. I just don't know what it is. Something about how.
All right, so this becomes our trivia question. Why are Diana's hydrangeas not blooming? I'm asking everybody.
That's interesting. I have some that bloom a lot, you know, more than you can believe.
Go ahead.
Diana, are you pruning them back either in the fall or early spring?
It would be in the fall that I have pruned them back, and then I think.
Is that all the way to the ground?
No, I have not. I did that the very first time, and all I got was green. Now I'm thinking of doing it again because I didn't do it for 2 years, and all I got was 3 blooms. I don't know. I just can't seem to. So I don't know what to do.
So what are you getting at there? What are you getting out there, Don?
Sean. His name is Sean.
Or Sean. Okay, I'm sorry. Well, Sean, what are you getting at?
Well, that's what I've always done. Is in the late fall after the flowers die back, I always go out and prune them all the way to the ground and put some compost on them.
I'll try that.
I get full blooms all over, you know, the summer and that.
So one of the things that can come into play that I thought Sean might have been bringing up is that when we say the word hydrangea, there's a lot of different plants. So there are many different sorts of hydrangeas, everything from climbing hydrangeas to, you know, the PJ almost tree-like hydrangeas, the everbloomers, all that kind of stuff. But one thing we have to be aware of with pruning is that you have to know when the flower buds are developed. So sometimes on certain hydrangeas, flower buds develop in the fall. Sometimes they develop in the spring. So imagine that the flower buds develop in the spring and you prune in the fall.
Then you're doing the right thing because they're coming up in the spring, right?
So what about if you prune in the fall on the flower buds that were already developed and you do prune, let's say you give them haircuts, you just do some pruning back.
Okay. Sometimes they have buds in the fall. There's certain canes that have buds, you know?
So the key to pruning is going to be not pruning off the flower buds.
Right.
For example, if you had a forsythia and you pruned them in the fall,
right,
you end up with no flowers. Because forsythia is going to be, their flower buds are going to be developed and on there in the fall. And if you then prune, give them a haircut, you won't have any flowers in the spring. So you have to be aware. So one thing I will say to you, you have to find out what kind of hydrangeas you have.
Okay, and I have, there are 3 kinds.
All right.
The kind that you have to prune, that you cut all the way down, and they only grow on new cane. There's the kind that grows on the, I guess it's last year's cane, right? And that one you don't prune, you don't prune it down in the fall. you wait until after it's bloomed and then you prune it, which is early fall, actually. Okay. So, and then they're the kind that have both of those, right? Right. So, and I can't figure out, I don't know. I'm going to try again this year. I haven't seen anything growing yet. But then I also have to take into consideration that our environment is not cold the way it used to be. Do you know what I'm saying?
Yeah, I'm not sure that that's affecting hydrangeas, but okay.
So I don't know. And I'm thinking that maybe it has both, some old and some new. So I don't know what to cut back half the time.
All right, so I guess what I would say is you have learned about the timing of the flower bud development and how that relates to pruning. So that's something that you have learned, and that's something that a person who is thinking about making pruning cuts on their hydrangea needs to keep in mind. What is the best time of year to prune for that particular type of hydrangea? The second most, so that's one of the most common problems why people don't get the bloom that they
Right.
Second most common problem that I would say that they don't bloom is that most hydrangeas require a good bit of sun. And here is one of the things that I have seen happen to a lot of people, not you necessarily, but a lot of people discover that their environment, their landscape, which they think is the same landscape that it was 5 years ago or the same landscape that it was 10 years ago, becomes a different landscape relative to shade because of trees. And so you're going to get reduced bloom on, and I'm not saying this is what's happening in your case. I'm saying that a common problem is that you end up with more and more shade and less bloom because you haven't really thought through the fact that there has been, I don't want you to get too
Okay, I know, but I don't think that's it. Okay.
Okay, all right.
Because I find that is my hydrangea, this one in particular, and I got it from Frank Bishop, as a matter of fact. He gave it to me. And I don't, it doesn't matter. And okay.
Hey, Diana.
But wait a minute, let me finish, because I planted this where I cut a tree down. So it does get light and it dries up. It is more susceptible to overlight. Do you know what I'm saying? When there is too much sun.
They wilt.
It wilts like you wouldn't believe, and I have to make sure that it's watered whenever it's too hot out. So
well, okay, so my answer to you is I'm not sure why you're not getting bloom.
What?
Those are two of the most common problems. Does somebody else have a comment?
Yeah, Gary did.
Diana, I have some hydrangeas. So one, if anyone wants them, they're welcome to them. I'm getting rid of them. Two, in this, I don't trim mine because I have the same question that you do, and I read something about this, and so I just don't trim them until in the spring, when half of the stalks are clearly dead and the other half are clearly alive.
Yeah.
Then I cut the stalks that I know are definitely dead and I use them to start fires. But I only do this in the spring after the leaves have just started.
Okay, and that's what I did this year.
And I have really big leaves, so I think I have macrophylla. And I can take a picture and show you mine. I can send you a picture of my hydrangea tomorrow so you can compare and see.
Okay, I don't think mine have any leaves left, but yes, but mine have large leaves too. So I don't know.
Mine did fall
off.
I think the answer in this case may be get some plants from Gary.
I have 1, 2, 3, 4 hydrangeas. Okay, the only one that works and I don't know. Okay, we're not going to discuss it anymore. I just have to keep experimenting. It's okay. Nobody's going to be able to figure it out but me and the hydrangea.
How about I send you some Rosa Sharon?
I know, I have plenty of those.
There's another.
I got that. So it's up.
Gary, do you have a question? Or Adam?
I do.
Go ahead.
Is OSU, has there been much study for the region around hemp growing and any diseases that it's localized here?
Well, I don't know the answer to that. I mean, there's part of the answer is we do have an extremely growing hemp production program. And the person who has really been involved in that used to be a program assistant in Geauga County many years ago when I started with Extension, but he then went to the OSU research station in, near Jackson. I've forgotten the name of it. Maybe it's in Jackson. And so Brad Bergerford is his name. He's a really great guy. So if you check out, I mean, I would guess if you Google OSU plus hemp plus Brad Bergerford or whatever, you would get some good information. I don't know how much we have delved into the disease problems that are occurring on hemp and on hops. Hops, actually, the hops plant is reasonably closely related, actually. Brad has done a lot with hops. He's done a good amount with hemp. So we do have a burgeoning OSU program. I'm not really that, I mean, I almost got involved in it because 2 years ago, I guess, 4 years ago, when I was first going to retire, somebody wanted me to become, since I was retiring, their marijuana consultant. So it was when everybody was going into the competition for who would be able to grow marijuana in Ohio, obviously related to hemp, and so they all had to have a specialist in various arenas, entomology and horticulture and plant pathology. So, they offered me a big chunk of money, and it turns out they didn't win the sweepstakes, and I already told them I wasn't going to do it anyway. But there are diseases. I guess I'm just not very well aware of which particular diseases. I mean, there's a powdery mildew that certainly gets on them. There's a Phytophthora. So Phytophthora is a water mold organism that used to be called a fungus that causes root and crown rot. And so you have to be very aware of that and having good soil drainage and good oxygen availability to the root system. But beyond that, I'm not really up on what are the specific problems people are finding in hemp production. But Brad would know that. So Brad Bergerford, he might be bergerford.1 at osu.edu. I kind of think he is. B-E-R-G-E-F-U-R-D, Bergerford. So Brad is probably up on that. I'm not up on the diseases of hemp.
Thank you.
All right. Aaron, do you have a question?
I will say that I get a lot of questions on that when I do horticulture and plant pathology in the prisons. That's kind of the first question. As people are kind of thinking, what am I going to do when I get out? It's just, unfortunately, that is kind of where everybody thinks there's going to be a lot of money in marijuana production. So they are always asking that question when they hear about the word horticulture, as you can imagine. But it's unfortunate.
Yes, it is. Aaron,
first thing that comes to mind.
Do you have any questions?
I have a lemon tree someone gave me, and I've started to have some issues with it. It's little tiny brown bugs, but I'm also realizing that symbiotic relationship, right? So it's not just the bugs. I also have a fungus. So I'm like getting, and it's an indoor plant, but I think this is pretty common.
Okay, so these little brown bugs that are stationary.
Yeah, they're scaling. They're on the stem. They're moving
Right.
about under the leaves. Yeah.
Right. So what you're seeing there are what are called scale insects.
Yeah.
And so the scale insects send their mouth parts in, and they suck nutrient from the plant. And then, as you have alluded to, they excrete that out the other end. And we have a nice, pleasant way we don't call it pee or poop. We call it honeydew.
Yep.
And the honeydew is just a sweet substance where the insect has used some of the sugar for their own metabolism. They used some of the proteins from the plant and fats from the plant, but basically they were using mostly sugars for the energy for the scale insect. And then they excrete some of the sugars out that they don't need. And then a fungus grows on that, which actually is not considered a plant pathogen. The fungus that lives on the honeydew, this excretion, secretion, the fungus is called a sooty mold fungus.
Yeah.
And the sooty mold fungus causes a sootiness. It does not directly infect the plant, because there's the thing that maybe I'll close with this. One of the things that we talk about when we talk about infectious plant diseases is we talk about the fact that there's a host-parasite relationship inside plant cells. That's not what is happening with the sooty mold fungi. They're simply living on that sugary substance. They're simply living on the honeydew. They're not living on the plant leaf, even though they look like they're living on the plant leaf. And you will have noticed if some of the honeydew falls on the saucer or on the wherever you have the plant, the sooty mold fungus will grow on that too. I mean, they're not growing on the saucer either. They're just growing on the honeydew excretion. So fungicides, for example, are beside the point. The issue is the insect that you would want to try to control. So you probably have San Jose scale or another scale that somehow got developed on that lemon tree when it was in the greenhouse, probably, or if you have a lot of house plants, then maybe you have an infestation that's spreading amongst your house plants. So those become, fairly significant problems in homes, and it just depends on how many of them there are and how willing you are.
It was near a bay leaf, so I had a bay leaf. So those two together were there, and I think it just ended up happening that they both sort of spread.
So if it's a limited number, you can just pick those off, and because what's going to happen is that the scale insect will mate, and the female scale then will produce little eggs underneath that underneath that little scaly covering. And then those little eggs will hatch, and then the crawlers will come out, and they'll crawl to other leaves on plants that are affected by that particular kind of scale, and then they'll increase the populations. And so picking them off and smashing them, dabbing them with rubbing alcohol can be helpful, or you can use insecticides. The problem with insecticides is there's only so much you're going to want to use inside your house.
How about neem oil? Is that neem and like water?
It's somewhat effective, sure. But as you can imagine, with that covering on that scale insect, it's kind of hard to penetrate. So they're historically difficult to control. The ideal way to control them is to time the period when the crawlers are out. In other words, when those eggs hatch, then insecticides easily control the crawlers, because the crawlers come out and then try to establish a new place to penetrate that tissue. And that's when they're very vulnerable to neem oil or whatever else you're using. It's just going to be difficult, not impossible, but oils can be very helpful. Along with, again, if you have a limited number of plants, just picking them off, smashing them. Real biological control, smashing them between the thumb and forefinger. Let me show you one thing here that I think is important, because it really is a fundamental thing to think about relative to plant disease, infectious plant disease, in terms of what goes on. So let's imagine that we have rose black spot, and so this is going to be diagrammatic, and so we're outside the actual scale. So this microscopic spore lands, so it's going to spread in some way. Part of the disease cycle is an insect bring it? Does the wind bring it? Does splashing water bring it? Typically, splashing water would bring a rose black spot spore to a rose leaf. So this is the rose leaf. So we have the cuticle on the upper leaf surface, the upper epidermis. We showed that earlier with that powdery mildew slide. Here's the lower leaf epidermis. Here's examples of how the gas exchange occurs. Carbon dioxide necessary for photosynthesis coming in. Water vapor is being lost through these stomates and the cells with those associated with it. Here's where photosynthesis is occurring. Here's where the vascular tissue links up to bring water and minerals, to bring water and minerals up through the flow from the xylem, and to take the food produced by photosynthesis down to the rest of the plant in the phloem. But so here's this representative leaf. And so what happens is, and with rose black spot, it's mostly splashing water, these spores arrive. So the fungal spore lands on the surface of the plant leaf, and it obviously is not red, and it's obviously not that big, but so it helps for us to see it here. Now, if the conditions are right, and remember we said with rose black spot, a key ingredient is the number of hours of leaf wetness. And if it happens to be a rose that's susceptible to black spot, and by the way, the fungus that causes rose black spot occurs only on roses, only on the genus Rosa. So it doesn't occur on anything else. We have some diseases called black spot, like elm black. It's a totally different deal. So the fungus that causes rose black spot, Diplocarpon rosei, has its own Latin binomial, only occurs on roses, and not on all roses, but on certain susceptible roses. So this fungal spore lands, has the right conditions of leaf moisture, and it germinates. So it germinates kind of like a seed germinates under the right conditions. Now then it's coming into contact with the plant, and the plant has various ways to try to deal with this. It has some constituent materials that help it resist this. That's its genetic capability of resistance. If that plant spore germinates and gets into the plant, there's some other reactions the plant can take But what happens if none of that works is that the fungus penetrates the plants, like we saw those Hostoria with the powdery mildew fungus. And this is why rose black spot is worse than powdery mildews typically, because it penetrates down past this epidermal layer of cells, penetrates down into the plant tissue, and then enzymatically degrades the constituents of the cell. That's crucial. It's not like an insect, like that scale insect. It's not like an insect sucking sap out of the plant. It's not like an insect sending toxins down into the plant. Literally, the fungal pathogen is enzymatically breaking down the constituents that are inside these little individual microscopic cells of the plant. And by doing so, they get their own food. So the fungus is getting its nutrition from feeding on the plant. And then ultimately over time, basically damaging and killing cells. And then you get a black spot, which would be the symptom. And then you get yellowing, which is a symptom. And you get defoliation, which is a symptom of that enzymatic breakdown and feeding on the plant by that fungal tissue. It's a host-parasite relationship inside those plant cells, like we have with human pathogens, a host-parasite relationship inside those cells. Viruses do it differently, but there are viral plant pathogens, and of course there are viral human pathogens, but it's a host-parasite relationship, which is so different from insects. It's not just a matter of seeing the insects, they're feeding, you kill them off, they get high enough numbers. You're actually, once you're in, we have very limited success with doing anything to that organism once it's inside the plant. So if you were to prevent disease with a fungicide, this is what you'd have to do. You'd have to apply a fungicide, and then when the spore lands and germinates and tries to get in, the fungicide kills the spore that's germinating. And that, again, is the whole problem with using fungicides. You have to know that an infection occurred. How do you know that? Well, you know, growers do some various things. They know what the environmental conditions are that are conducive to disease. So when they see those conditions occur, that's when they would make protective fungicide applications, protectant fungicide applications. Sometimes that's very elaborate. They'll have hygrothermographs out in an orchard or in a potato field that are registering things like temperature and relative humidity and moisture. And so then they are able to predict that an infection would occur. We have some fungicides that are called eradicants and curatives, which sound like that's opposite what I'm telling you about the need for protection. But those eradicant and curatives are within 24 to 72 hours of infection. And when we talk, for example, of rose black spot, before you see rose black spot, when you see this symptom, that's about 10 to 14 days after that initial infection. So 24 to 72 hours, 1 to 3 days, 1 to 3 days after 1 to 3 days after penetration, you're not going to see anything. So in other words, that's the dilemma with plant disease control with fungicides. You have to anticipate that there was an infection. So that could mean highly targeted work with understanding what the environmental conditions were, or a schedule that you know that's based on, okay, we need to apply as soon as the leaves come out because we know that. So you may be making applications that don't need to be made, or to use one of these eradicants or curative, but you have to use those within 1 to 3. So, you know, you'd say, oh my God, we must have had an infection period last night. Let's make an application now. But of course, that's very difficult to do as a homeowner. So that's why diseases are difficult to control through control of the pathogen in this way. That's why we'd love for people to simply plant resistant varieties. So for many diseases, we have resistant varieties that then won't get that particular disease. We have roses that don't get black spot. Now, the problem with roses, unfortunately, for the rose grower, for Chris, is that the big thing looming right now is rose rosette virus, because we don't have any controls for rose rosette virus. We do not have viricides. We do not have roses that appear to be reliably resistant to rose rosette virus. So it's becoming more and more problematical to feel good about having a big, large rose garden. And people like Cedar Point and places like that, that had large rose gardens as ways to draw people in, it's getting to be very difficult because we don't have any management approaches. We have some. I mean, we have areophyid mites are known to be the organism that vectors the pathogen. So you can have a very, very intensive areophyid mite control program, but it's tough. I talked to, the last time I was with my class in New York City, we swung over to the Brooklyn Botanic Garden. We walked over and talked to the curator of the Rose Garden. I said, well, how are things going? Massive rose garden at the Brooklyn Botanic Garden. I said, how are things going? And he said, well, all right. And I said, you know, he says, how do you manage various diseases? Rose black spot, powdery mildew. He said, what about rose rosette? And he said, well, he said, I got to tell you, I don't know how many more years I can do this. He'd been doing it for 25 years. He said, I've just simply seen too much death. It was a really sad story. I've seen too much death. And so what's happening is that they're basically, you know, they want to have the rose garden. It's a big, well-known aspect of the Brooklyn Botanic Garden, but he's losing, you know, 10, 15, 20, 25% of his roses every year. And they don't want him to just decide to, you know, create something that isn't a rose garden monoculture. They want him to keep, you know, this is a known thing. We got to have the rose garden. We got to have the rose garden. But, you know, they don't all die at once, but he's just tired of having to remove so many plants year after year after year. And I mean, I guess you can just look at it as, okay, you know, we're only going to have a rose for 3 or 4 or 5 years. And we just have to realize the fact that we're not going to have roses that stay very long. And it's not that every rose garden is that way, but for example, at Worcester, at the Ohio Agricultural Research and Development Center, which is now called OSU Worcester, you'll notice if you love to come to the OSU Rose Garden of Legend and Romance that it isn't there anymore. So we had a lot of rose rosette virus, and finally the decision was made at the Arboretum to take them out. And so now it is trial gardens for coneflowers, and now hydrangeas are being put in. So they just have not been able to maintain that rose garden there. It doesn't mean that Chris can't have roses. It doesn't mean that you can't have roses, but it is interesting. I did make a trip to New York City. I go 3 or 4 times, well, much more than that now, because my daughter lives there with their son and her husband. And I have made a couple of trips this year at various times when Ohioans were allowed in. And I was always impressed by the fact that somehow they had gotten away. And I talked to the arborist and the horticulturalist. They had planted literally, I don't know, it had to be in the tens of thousands of shrub roses along the Hudson River Parkway area in, you know, mid to downtown on the eastern side by the Hudson River, the western side of the Hudson River, of roses and beautiful, beautiful things. And I'd noticed for the last 4 or 5 years, my God, they're fantastic. They're beautiful. They're wonderful. They're lovely. How have they gotten away with it? I noticed the last time I went to New York that most of them are dead. So rosette is the real killer. And it's because we don't have good host resistance. We don't have a good way to control the pathogen. We can theoretically control the vectors, but they're not going to be having an intensive miticide spray program for their outdoor roses in New York City. And, you know, it's just, and the environment conducive to disease is present. So that's how it all works with plant disease.
Thank you
Hey,
very
Jim.
much.
Thank
Yeah.
you.
What's Worcester doing with coneflowers? What species are they trying out?
Well, that's a good question. So a really good guy that's the new curator there is Jason Vale. And so Jason and Paul Snyder works with Jason. They're just crackerjack, good horticulturalists. And so this was their second year for the coneflower trials. I believe they had, first of all, I can't directly answer your question, but I'll try to do as much as I can around the question. I want to say 64, although I think it actually was more taxa than that. And they maybe replicated them a couple times throughout the plot. So they had a wide range. I don't know how deep they went into species. You know, so there's different, I don't know how many, and you may know better than I, how many different species of echinacea there are. So they had multiple species. They had a lot of the different cultivars. They had double bloomers. They had a whole wide range. So they had a lot of them, but how many different species they had, I cannot totally answer. Purple cone, you know, all the different coneflowers.
Do they like to talk about it with strangers that call them?
Oh, absolutely. Oh my God, that's what they're all about.
Okay.
And they're really hoping next year. So next year will be the 3rd year. They'll probably do 4 years and then they'll move on to something else. But next year, I think their real goal is to start sorting out. So the diseases of coneflower, I mean, you have powdery mildew. you have a really serious problem known as aster yellows. So, aster yellows is caused by an organism called a mycoplasma. It's kind of in between, I mean, it's not really, this isn't exactly true, but it's kind of in terms of size, it's in between a bacterium, which you can at least see with a light microscope, and a virus, which you can't, you have to have electron microscope. So in between that is a mycoplasma-like organism, which is now called, it used to be called a mycoplasma-like organism because mycoplasmas occur in people, not plants. And then they eventually started to call it a phytoplasma, so phyto for plant. It's now classified as a type of bacteria, but it's a very tiny bacterium, in a wall-less bacterium that is, again, not, you can't see it with any kind of regular microscope. It's difficult, but aster yellows is a real fundamental problem. So aster yellows, you have anthracnose leaf spot, you have alternaria leaf spot. You have some phytophthora that's occurring on the stems and getting some dieback in some cases. They haven't had a lot of that, but they have said some of that. And then, of course, you have Japanese beetles from the insect side. You have Japanese beetles. You have different kinds of mites. You have an areophyid mite that causes symptoms very similar to aster yellows. You have, some other insects. So I think next year they're going to kind of start to roll out a lot of evaluations for those aspects. But so far, their evaluations, which they've done, which are quite extensive, are on, how the plants are holding up. It's a pretty good test because certainly the first year was very dry. So, here they had them out there in full sun in a very tough environment with not very much moisture. They're not irrigated. So it was a good test of how well they would do in an average garden. And so, but they really looked very, very beautiful until late in the season, of course. So yeah, they have the details on it. And both Jason Veil, V-E-I-L, I can tell you their email address if you let me see what I can do if I pop them up, but Veil.something. I think it's Veil.81. And then Paul Snyder.1million or whatever. But yeah,
I can get the address for you for
yeah.
them.
So Lola can get that, but so they're really good guys. Jason's the curator of the Arboretum, and Paul is the educational director and assistant curator. They're fantastic horticulturists, and they really are doing a nice job with it. As
Seacrest Arboretum.
Schultzman,
Yes.
what?
Seacrest Arboretum.
Correct.
Got them.
All right,
Cool.
great. Well, good job.
Thank you.
All right,
Yep.
thank you. Okay, we're going to go ahead on and wrap it up. I want to thank everybody for hanging in there tonight. And I want you all to remember that Jim will be back again with us to close out our calendar year on December 10th, which is another Thursday at 7 p.m. Come with your questions and be prepared to be wild with information from Jim.
Let me give you two final things here.
Go ahead.
That I did. Just, you always got to have a little bit of biology.
Go ahead, Jim.
Yeah.
I recommend this book, I Contain Multitudes, The Microbes Within Us and a Grander View of Life. Every person aerializes around 37 million microbes per hour.
That's scary.
So think about that for a minute. That's why we're wearing masks, ladies and gentlemen. And then this really is a good quote from Rene Dubos, which is really part of the modern synthesis of evolution that we're in the 50s and 60s. Modern man believes that he has achieved almost complete mastery over the natural forces which molded his evolution in the past, and that he can now control his biological and cultural destiny. But this may be a delusion, which we certainly know in our age of COVID-19. Like all living things, he is part of an immensely complex ecological system and is bound to all its components by innumerable links at some unpredictable time and in some unforeseeable manner. Boy, are we living this. Nature shall strike back. And then we go to Darwin. There is a grandeur in this view of life from so simple a beginning, endless forms, including pathogens, most beautiful and most wonderful, have been and are being evolved, and not to our benefit, but certainly part of nature. One of those things that has evolved is a novel coronavirus that we're now living with. All right.
Let's give him a hand. Thank you, Jim, so much.
You bet.
Happy
Thank
you, Jim.
December 10th, we'll be back for to close
All
out
right.
the year.
All
PAO.
right, sounds great.
All right.
Everybody
All right.
have a great evening.
See ya.
Yeah, you guys do.
Thank you.
Thanks all.
See you,
Have a
Lola.
great